Friends, welcome to Biology Made Easy. Today in this video, we will discuss nucleus. So, as all of you are aware, that a nucleus is the basic difference between eukaryotic and prokaryotic cells. In eukaryotic cells, there is a true nucleus, but in case of prokaryotes, that a true nucleus is absent. What is the meaning of this true nucleus? That means when the nuclear materials, that is the genetic material, DNA and RNA, they are surrounded by a nuclear membrane, then that is known as true nucleus. But when the genetic material is not surrounded by any nuclear membrane, that is simply known as a nucleoid. That is found in case of prokaryotes. So this is the basic difference between the eukaryotic cells and prokaryotic cells. In eukaryotic cells, the nucleus is considered to be the largest organelle in comparison to all other organelles like plastids, mitochondria, endoplasmic reticulum, Golgi body. This is the largest organelle, and this nucleus is the permanent storehouse of all the genetic material. That is why it is also known as that is genetic repository. This is also known as genetic repository. That means it is the storehouse of all the genetic information that is present. Now, when the nucleus is so important, who has first discovered it? Actually, it was first discovered by Antony van Leeuwenhoek in red corpuscles of fish, but he was unable to study the details. And ultimately, it was the Scottish botanist Robert Brown. He discovered it from the orchid cells. So initially, it was discovered by Antony van Leeuwenhoek in red corpuscles of fish, but actually it was by the Scottish botanist that is Robert Brown. Robert Brown in 831 he studied the details from the nucleus of the orchid cell. Now, usually this nucleus is present in all eukaryotic cells, with few exceptions. So, what are the exceptions? Usually, the exceptions are that is mature RBC. Mature RBC in case of the animal cells, and the mature sieve tubes. Mature sieve tubes in case of plants. So only in case of mature RBC and mature sieve tubes they are absent. That means initially when the RBC was not mature, or you can tell immature, the nucleus was present, but after maturation it disappeared. Similarly, when the sieve tube is formed, the nucleus is present, but when it becomes mature, then it is completely absent, because it will help in the translocation of food materials in case of plants. But if from a cell the nucleus is removed, then the cell starts functioning, or it stops functioning. That means nucleus is the most important part of the cell. You can tell that it is the heart of the cell. It is the engine of the cell. If it is removed, then the cell stops, it ceases functioning. So what is the importance of this nucleus that was studied by one scientist, that is Hammerling, J. Hammerling in 1953, he studied the importance of the nucleus in acetabularia. Acetabularia is unicellular. It is one algal cell. Though it is unicellular, it is a very big cell, around 10 centimeter in height. In acetabularia, there are usually two types of species. One is Acetabularia mediterranea, and other is Acetabularia crenulata. Now, what is the difference? In case of Mediterranea, the cap is like this ray-shaped, while here the cap is star-shaped. It is star-shaped, and the remaining parts, this is the rhizoid part. And the nucleus always lies in the rhizoid part. That means here the nucleus is present in a rhizoid. So what Hammerling did, he made a grafting experiment. When this is cut, that means in one part the cap is there in the, which is the enucleate, and this is the nucleated part. That is, from this nucleated part, the entire cell originated including the cap, while from this cap portion which lacks nucleus, that degenerates. And the same thing also happened here. That means the cap region degenerated, while from the nucleated rhizoid portion the entire cap regenerated. So from this it is clear that a nucleus, it gives rise to the entire cell structure. Then he made a grafting experiment. In the grafting experiment, the two were combined together. That means both the nuclei, they were combined together. Due to the grafting of these two were combined together. And that means both the nuclei are present in one. Both are present in one. And it was found that this intermediate one, which is formed due to the grafting of the two, both the Mediterranean and crenulata, it gave rise to the intermediate type, that is intermediate to both this ray-shaped and the star-shaped cap, intermediate type. So from this it is clear that a nucleus is the controller of all activities of this cell. It is the heart of this cell. That is the importance of this nucleus. Now, if we will discuss the other aspects of the nucleus, the next point that we will discuss is number. That means how many nuclei are present in each cell. Usually, majority of the cells, they are uninucleate. That means in each cell there is a single nucleus. This is very common. But there are some cases you will find where they are binucleate. Binucleate means there are two nuclei in each cell. And the common example is Paramecium. Paramecium caudatum. You will find in each cell there are two are there. There are still examples where you will find multinucleate conditions. Multinucleate condition. Now for the multinucleate condition, we can use two synonyms. What are the two synonyms? We can use the two synonyms. We can use the word Coenocyte and Syncytium. These are the two terms. Usually the Coenocyte term is applied in case of plants. And when usually we call this the Coenocyte, when there is a pre-nuclear mitosis. Pre-nuclear mitosis. That means the nucleus divides without cytokinesis. So in that case, what happens? So one multinucleate condition is formed. That means there is mitosis, but there is no cytokinesis. So what are the examples? The common examples are, one example you can tell the liquid endosperm of coconut. Liquid endosperm of coconut. This is a common example. Then the other examples also you will find in case of fungi, that is a rhizopus. And from the algae, the example is Vaucaria. Now come to the Syncytium. The Syncytium, this term is usually applied mostly to the animal cells. But here these are not formed due to the free nuclear division. They are formed by the fusion of different uninucleate cells. Suppose there are so many uninucleate cells. Uninucleate cells, all are uninucleate cells. Now all of them will fuse together. They will fuse together. So now one multinucleate condition appears. So this is the basic difference between Coenocyte and Syncytium. So here the common example of this Syncytium, it is usually you will find in case of the striated muscles. Example, striated muscles. Then so we will find also in case of opalina and also in the epidermal cells of ascaris. Epidermal cells of ascaris. So this is regarding the number. The next is the position in the plant body. Sorry, position in the cell. What is the position in the cell? Usually in most of these cells, and particularly the animal cells, it is almost central in the position. So it is in case of animal cells. In animal cells, it is usually present in the center. But in the case of adipose cells, of animals, it is usually peripheral. This is the difference in adipose cells. But in case of plant cells, it is always peripheral. In case of plant cells, it is always peripheral because it is pushed by the vacuole. Peripheral because pushed by the central vacuole. So this is the position. Then the next thing that we'll discuss is the shape. The shape and size. Now if we will discuss the shape, usually the nucleus doesn't have a definite shape. It may be polygonal, or you can it may be oval, round, spherical, or cuboidal, or cylindrical. Any type of shape is there, cylindrical. But in case of plants, since it is pushed by the vacuole, in case of plants it is usually lens shaped. In case of plants, it is usually lens shaped because it is pushed by the vacuole. So this is regarding shape. Now come to the size. The size of the nucleus. Usually the size of the nucleus also varies from cell to cell. It depends upon the number of chromosomes, because the number of chromosomes varies between haploid cell, diploid cell and polyploid cell. So depending upon how many chromosomes are there, the size differs in haploid, diploid and polyploid cells. That is one aspect. Another important aspect is the size of the nucleus depends upon the mass of the that is the nucleoplasmic mass of the nucleus and the cytoplasmic mass of the cell. That means we can tell that is the nucleus divided by cytoplasm. That means the mass of nucleus and the mass of the cytoplasm, always a definite ratio is maintained in almost all cells. So depending upon the cytoplasmic mass, the size of the nucleus will also vary. Otherwise, it also may vary depending upon the number of chromosomes, as I already have told it, depending upon haploid, diploid and polyploid cells. So this is the size. Now if we'll, the next thing that we'll discuss about the chemical composition. Then the next that we will discuss is chemical composition. In chemical composition, 80% of the nucleus consists of only proteins, which are again of two types, maybe histones or non-histones. That means 80% of proteins are there. Out of this 80% proteins, 15% proteins are histone proteins or histones, and 65% are non-histones. Non-histones and enzymes. Non-histone proteins and enzymes are also proteins. So 15% histone proteins and 65% non-histone proteins and enzymes. That is mostly evident thing. Then we'll get 9 to 12% DNA, 5% RNA, then 3% lipids, and there are some minerals in traces. So the different minerals in traces, the minerals are usually calcium, potassium, sodium, magnesium, phosphorus. All these are present in traces because they play also very important role in the functioning of the nucleus. So these are the different aspects of the nucleus. Now let us start discussing about the nuclear envelope or the detailed structure of the nucleus, where we will discuss the nuclear envelope. Now the nucleus consists of mainly 4 parts. One is a nuclear envelope or Karyotheca. Then the second is nucleoplasm or nuclear shaft, or we can call it a karyoleaf. The third one is chromatin, and the fourth one is nucleolus. That means let us discuss the structure. So it has usually 4 parts. Number 1 is the nuclear membrane. Nuclear membrane, or we call it as Karyotheca. Karyotheca. Then the second one is nucleoplasm. Nucleoplasm or karyoleaf, or this is also known as nuclear shaft. And the third one is chromatin, and the fourth one is nucleolus. We'll discuss one by one. First, let us discuss this nuclear membrane or Karyotheca. Usually, each nucleus has a double membrane structure. One double membrane structure is there. So this is the inner membrane, and then there is a outer membrane. That means there are 2 membranes. Now the outer membrane, that may be that is usually connected with this endoplasmic reticulum. At some places it may be connected with the endoplasmic reticulum, and this is usually rough endoplasmic reticulum. And in addition to that, the outer nuclear membrane, it has also ribosomes. Outer membrane. That means the nucleus is surrounded by a double membrane structure, and out of this outer membrane, this is the outer membrane. Now the outer membrane is usually rough because of the presence of the ribosomes. It is rough because of the presence of ribosomes. And the outer membrane may be connected with the endoplasmic reticulum. That means this is directly connected with the endoplasmic reticulum at certain places. while the inner membrane is smooth. The inner membrane is smooth, and the thickness of these membranes usually varies between 60 to 90 angstrom. And the space between the inner and outer membrane, this is known as perimitochondrial space, perinuclear space. This is known as perinuclear space, and it is about 100 to 500 angstrom in width, this space. Now, it is a double membrane. The outer membrane is rough and connected to the endoplasmic reticulum. The inner membrane is smooth, and both the inner and outer membrane, they are free from each other, except at this point of the pores, that is where it is continuous. It is a continuous. And the nuclear membrane is not uninterrupted. It is interrupted by several pores. These are the different pores, and these are known as nuclear pores. Nuclear pores. Usually the number of nuclear pores varies from 1,000 to 10,000 per nucleus. Their number varies. And the nuclear pores, it connects between the nucleoplasm which is present inside the nucleus with the outer cytoplasm. But what is the structure of this nuclear pore? That was for a long time it was debating, because somebody told that there is a septum, so there is a plug, or there is a diaphragm, like that. But the latest one is this nucleus, the structure of the nuclear pore. The structure of the nuclear pore is an octagonal orifice. Octagonal orifice. That means it consists of two rings. Which are known as annuli. And they are connected by means of spokes to a central core-like structure. That means if you draw the diagram, then it will look like this. Suppose this is one ring, and this is another ring, and there is one central pore-like structure. Now, in this side there will be eight rings, and this side also there will be 8 rings, and there is one central one. Now, these are connected to the central one. They are all connected to the central one. Now, you see how the whole thing looks like. The whole thing looks like a dumbbell structure. That means on this side the outer membranes are present. That means this is one outer membrane. This is the outer one. This is the inner one on one side, and this is the other side. That means these two are outer membranes, and these two are inner membranes. And both in the outer side there is a ring, 8 rings, which gives rise to one annulus, and in the inner side there is 8 rings that gives rise to another annulus, and both of them are connected by means of spokes. These are the spokes to the central one, and this the diameter of this central one is usually, it varies from 50 to 100 nanometer. That means the width of this pore is 50 to 100 nanometer. That means whatever substance will enter in through this nuclear pore, that means anything that will go inside or outside, that has to pass through this nuclear pore. Now, this structure is known as the octagonal orifice, and this is the latest structure that is accepted at present. That means 2 rings are there, and they are connected to the central pore by means of this. Now, in this pore, there is one important protein that is known as nucleoplasmin, and that nucleoplasmin that facilitates the to and fro movement of substances. So there is one protein known as a nucleoplasmin. This is very important from examination point of view. Nucleoplasmin protein is present in this pore that regulates the movement of substances. So what are the different substances that have to and fro movement? Inside there, the DNA is there, and the DNA will transcribe the different types of RNA, mRNA, tRNA, rRNA. They have to go out. Then here the nucleus is there. Nucleus is the site of synthesis of ribosomes. The bigger unit, smaller unit, that is 60S and 40S, they will also go out. The different types of minerals and substance, they will also go out. So they will go out only through this nuclear pore. That will say, through this nuclear pore, the ribosomal subunits, then the different types of RNA and other substances, they move from this nucleus to the cytoplasm. But from cytoplasm, different substances come inside. Again, the different types of RNA which are formed inside the nucleus, the RNA are very macromolecules and they are hydrophilic in nature. So since they are hydrophilic, they cannot pass through this nuclear membrane, because the nuclear membranes are just like the unit membranes, lipoprotein membranes. That is why the pores are essential for the movement of the different RNA molecules. So that is the important function of this pore, through which the different substances they move to and fro, and they are regulated by this nucleoplasmin. And another important thing, once the ribosomes are formed, both smaller and bigger, they combine together, or the ribosomes and they are activated, they cannot come inside because the pore will not allow them. So this is the important function of that nucleoplasmin protein which is present in the nuclear pore. So this is about the different aspects of the nuclear membrane or karyotheca, its structure and its importance. The nuclear membranes, it maintains the shape of the nucleus, and also it facilitates the to and fro movement of the substances to and fro. That means from the nucleoplasm to cytoplasm and vice versa. Then the remaining three parts, that is nucleoplasm, chromatin and nucleus, these things we will discuss in the next video. Thank you very much for watching the video. Wish you a very bright future. God bless you.